We're going to discuss about life cycle of bacteriophage. So, is bacteriophage basically complete the life cycle in two ways? So, if I can write here, the bacteriophage complete life cycle in two ways. We complete in two ways. So now let's say the first we can say way is basically the Lytic cycle, and second is lysogenic cycle. Second is lysogenic cycle. Now remember this Lytic cycle is also known as master and slave cycle. Lytic cycle is also known as master and slave relationship. And lysogenic cycle is known as, we can say, lysogenic cycle is known as host and ghost relationship. Host and ghost relationship. Fine. So now let's see how this is. Basically, these life cycles. Now, if we talk about here Lytic cycles, Lytic cycles include T event phases, which will be T2, T4 to 6, as we have discussed with event phases in the previous videos. So Lytic cycles basically include what? Lytic cycles include T phases. I guess T event phases, and those are basically T4 and T6. Now from these all we can say T4 is basically what? T4 is double stranded DNA. Remember this T4 is double stranded DNA. Fine. Now why the Lytic cycle includes these T phases, T even phases? Just because here we have these have well developed fibers, and in the Lytic cycle's whole work is of fibers, they have well developed fibers. Okay, so I have this is clear to you guys. Now we should move with the life cycle, Lytic cycle. So yep. So Lytic cycle is basically, as we all know. Okay, let me before telling you about lysogenic cycle. So here basically lysogenic cycle basically include what? Lysogenic cycle includes we get the lambda phage. Includes lambda phages which are basically absent or they have not growth fibers. Absent or no fiber growth. Okay, so these are used in what? These are used in basically lysogenic cycle. So now let's see about the Lytic cycle. So we are going to discuss about the Lytic cycle. So first in this Lytic cycle we have what? First is this cycle is defined by the bacteriophage yeast, or this basically processes involved. Lytic cycle is involved the lysis of cell or killing them. Lysis of cell or killing them. So now this cycle is also known as I told you, master and slave cycle. Why master and slave cycle? As the master include what? Master include bacteriophage, and we can say slave include bacteria. So this is why this is known as master and slave cycle. Now this basically phage will destroy, this bacteriophage will destroy the bacteria and known as virulent or lytic phase. So that phase is known as lytic phase. Phage destroy the, you can say base phage destroy phage destroy the bacteria. Phage basically destroy the bacteria and we can synthesize own proteins. Right? So now let's see about this So yeah, this phase is basically completed into following steps. So following steps, if you can write here. So first step is basically attachment or adsorption. Attachment or adsorption. One second phase is genome penetration. Genome penetration. Third phase we have replication or biosynthesis. Replication and biosynthesis. Fourth phase we have basically assembly. Last phase is basically release of new phages. Release of new phages. So I have this clear. Now we should discuss each phase in detail. So yeah, we are going with the first phase, which is attachment and adsorption. Okay, so in this first phase, in this attachment, what will happen? So now in the first phase, the bacteriophage are basically attached on the surface of bacteria, as this is basically the bacteria and this is the surface of bacteria, and these are the bacteriophage which will attach on the surface of bacteria. Okay, so first is bacteriophage attach on surface of bacteria. Okay, when this is attached, so with the help of the tail fibers, as the bacteriophage contain the tail fibers, so yeah, with the help of these tail fibers, this will attach. Okay, now tail fibers basically provide the strong we can say attachment on the bacterial receptor site. Here we have this bacterial cell wall, and here it include what? It include the receptors. So basically bacteriophage tail fibers attach to these receptors. So attach strongly to receptor of bacteria. Okay, to receptor of bacteria, because what? Because tail fibers also contain the we can say receptor binding proteins. So those proteins are binding to the surface of the bacteria with the receptors. Okay, I have this, we can say this is now binded, easily attached on the surface of bacteria. Now we should go with the second part, that is basically that is genome replication. Genome penetration. Okay, so what will happen in the genome penetration? So basically, as the virus was attached, after the attachment, the bacteria base plate, and the bacteria contain the base plate. If you have not watched the structure video, you can watch that. So you can say after attachment, bacteriophage contain what? Bacteriophage contain base plate, which contain bacteriophage base plate basically contain pin. Now it contain pins. Now these are the pins which are rectitable, which I told you, like it can be easily removed or it can easily attached. Okay, so they contain the pins, and these pins basically what? These pins create enzyme which is lysozyme. Lysozyme. Now what will the help of this lysozyme? So basically lysozyme is responsible. Lysozyme is responsible for doing the holes in the wall of bacteria. Responsible for holes in bacteria. So when the holes are done in the bacteria, so as I told you in the structure, there was one contractile sheet or contractile protein. We have contractile. So this contractile sheet is basically contracted. When this is contracted, so the bacteriophage DNA is injected into the bacteria. Bacteriophage DNA enter in bacteria. So when this bacteriophage DNA is entered in bacteria, but only the DNA will be we can say injected only. Baki the rest part, which is the proteinase part, will remain outside, and that part is known as ghost. So if I can draw here the rough diagram, so you can see here when if I have this bacteriophage wall, okay, and here I have here we can say bacteriophage. Okay, this is what I have drawn the rough diagram. So when this is attached, what it will release physically, here you can see the DNA will come here. DNA is the DNA of bacteriophage, and this is the bacteria DNA. Now how it come? By the contractile sheet. When it contracted, the DNA come inside of bacteriophage, and by doing the last like this, as we were having the pins. Pins contain what? Pins basically contain the enzymes. So it was the lysis of these are basically the we can say holes which are done by the lysozyme, through which by the contracting of contractile sheet the DNA of bacteriophage come inside the DNA. Okay, I hope this is clear to you guys. Now we should go with basically the third part, which is replication or biosynthesis. So here so if I talk about this replication or biosynthesis phase, so what will happen in this phase? In this phase, basically the bacteriophage DNA will destroy the bacterial DNA. As we have both the DNA here in the bacteria, so bacteriophage will destroy DNA. Destroy bacteria DNA, and when this bacteria DNA is destroyed, now we can say bacteria contain the metabolic machinery. So now the bacterial metabolic machinery is basically directed by the phage. Now phage will do whatever the phage want, क्योंकि now bacteria's DNA is destroyed. Host machinery directed by phage. Okay, as this is host machine is directed by the phage. Now what phage will do? Phage will basically synthesize their own structure. Now structure include capsid, protein, tail fibers, base plate. So we can say phage. We can say synthesize structure by using host cell machinery structure, or you can say components. So when these components are structured, and we can say double stranded DNA, which was what? Which was the T4, which was bacteriophage genome, and this will be also replicated. Also replicated. Now if I can give you here one example. So yep, here we have example of E. coli bacteria. Here we have E. coli. So what will E. coli is basically same as bacteriophage. Like bacteriophage are host specific, they will only kill the host specific. Like bacteriophage will attack on the bacteria, not any other thing. So their host is fixed. So yeah, E. coli, which contain basically E. coli phages, which contain coliphages, and these are basically these infect the one species, infect only the single species, or we can say these are also the specific to the species, but it can infect several different strengths. Now strengths are basically the several, we can say, parts. So those strengths can be infected, but it will infect only the single species like the bacteria. Okay, so I hope that it is clear to you guys. Now we should go with the part 4, which okay, in the next video we'll go with the part 4, which is assembly. Till that, bye bye. Chalo. So in this assembly, what will happen, as we can say, assembly simply means assemble. Each component will be assembled, like when the structured components are formed by using the whole self machinery. Structural component are formed. Okay, so now they are basically assembled. Now when they will assemble, they will make the assembly, their structure will be combined, they form a virion. After assemble, form virion. Now what is virion here? So virion is basically, we can say, complete infectious mature, we can say, virus particle. Complete infectious mature virus particle. Fine. So now let's see about here. We can say, let's see here if we have what the bacteriophage, we can say. So when DNA synthesized, now here we have phages only. So now these phages are basically assembled to each other, and their structure is assembled. When this is assembled, so the last part is basically what? Last part is lysis, release new phage. Release new phage. So now let's discuss that last part. Now these new pages which are formed, now those will be released. So yep, here you can see, as we have basically, when these new pages are formed here, these are, so now they will attached to wall of the bacteria through inside, and when they will attach, that means their tail fibers will be attached to the bacteria surface, so they will release enzyme which is lysozyme. And what was the work of lysozyme? Lysozyme basically do the holes in the cells. So when the holes will be present in the cells, so these fibers will be, new phages will be released. So if I can draw here how these are released, so here you can see. So this is how, by doing the lysis, these will phage will be released, and they will again infect another bacteria. So new phages released. So I hope till that it is clear to you this lytic cycle. In the next video, we're gonna discuss about lysogenic cycle. Till that, bye bye.